that we have 1,000 plants in this room right here, right now. Yes, especially after yesterday's cup plant talk, I don't think we'll be very surprised to find if there are more than 1,000 forms of leaves or floral organs. While we all would like to celebrate this seemingly infinite variety of lateral organ forms in plants, I always get also very excited by another aspect underlying this diversity, that there aren't 1,000 ways to make them. There's basically just one. The initial processes of producing a lateral organ is very similar among all plants, where we have groups of stem cells in the meristem giving rise to lateral organ primordium. As soon as the primordium is physically distinct from a meristem, it spontaneously acquires one axis, where cells adjacent to the meristem get the adaxial identity, and cells further away from the meristem get the abaxial identity. This adaxial-abaxial axis is very important because many of the final forms of a lateral organ is organized along it. Every organ has it. So maybe it's not really a surprise either that the genetic programs governing these two domains are very conserved as well. When major players of either party is missing, that will lead to the abaxial or adaxialization of the organ. For instance, here we have a classic example of a snapdragon leaf that's a flat wild-type leaf become abaxialized. So this phenotype got people very curious, because it seems like abaxialized or adaxialized organs don't have lamina. They're radial, they're needle-like. Closer examination of some chimeric mutants revealed that lamina growth only occurred at the edges of some sectors that had both adaxial-abaxial identity. So this observation led to a very important discovery in plant, important theory in plant development, that juxtaposition, the correct establishment and positioning of adaxial-abaxial identities leads to lamina growth. Over the years, this theory has been proven to be correct. In fact, many of the complex lateral organ forms we see in nature are due to the modification of the boundaries of adaxial-abaxial domains. So this is super neat. However, if you think a little bit more about this, things could be confusing. Juxtaposition of adaxial-abaxial identities leads to lamina growth. This is stating as if lamina growth is a thing, just one thing. As long as the domains are correctly there, cells on the boundary will just proliferate and expand altogether as a whole and make lamina. But we also know for sure that cells with different identities most likely have very different developmental trajectories. Even for a simple flat leaf, the inner anatomy show clear distinction between these two domains. So it seems like there's something more in between identity and lamina growth. Something, for instance, the identity program triggers some proliferation program, and the coordination between them give rise to lamina growth. Well, here comes the surprise. We basically know nothing about how cell proliferation is controlled in an identity-specific manner. But the brighter side is, we do know a fair number of genes that controls lamina growth as a whole. And among them, we picked the gene JAGGED and nubbing and conducted a typical reverse genetics in our system. So the gene JAGGED and nubbing are recent paralogs, first identified in our Arabidopsis, of course, both encoding transcription factor. They work redundantly to build anther wall, carpal wall, but JAGGED is the one playing a major role in all flora organs. When it's missing, the sepals and petals in particular will have defects in the distal part with jagged margin, and hence the gene name. The reason to see this phenotype is due to the fact that JAGGED is involved in the transition between cell proliferation and cell expansion, which is a very attractive aspect to us because our system, Aquilegia, the beautiful columbi flower, have some dramatic flora features, and we want to know how cell proliferation and expansion are controlled to build them. Aquilegia flowers belong to the buttercup family in the lower U diploid, so it can provide some kind of midpoint data to what's already known in the core U diploid and monocot. The flower is very interesting. For instance, it has this very elongated petal spur, 3D structure, and a fifth type of flora organ. Unfortunately, to tell you how fantastic they are is not the main point of my talk today. I hope you can join me in my talk in Botany 2018 for more information. Anyway, jagged Aquilegia reverse genetics. So I built a phylogenetic tree, identified the correct ortholog. We only have one copy. The jagged nubbing is probably resulted from the Brassicaceae specific duplication. Jagged in Aquilegia is expressed very broadly. That can be found in the shoot apex, flora meristem, lateral root primordia, and all lateral organ primordia, such as the leaf here and different flora organs, as well as organs undergoing rapid growth, such as this little petal here and the leaf. Subsequently, I tried to knock down the expression of jagged via VIX, virus-induced gene silencing. I'm pretty sure many of you here knows about VIX, so you must know some naughty feature about VIX, such as the silencing is not controllable at all. It could be patchy, it could be temporarily variable. Yeah, I see some nodding here. So usually these are mentioned as downsides of jagged, VIX when compared to stable transgenic lines. But you will see how they help me in my study here. So we have wild type, VIX control with a pigmentation gene. The first thing we noticed is that our flower has showed a whole series of defects in the distal part, ranging from the jagged margin of petals and sepals to reduced or even absence of the petal spur, as well as the anther development. Secondly, we saw many of our flowers don't have many flora organs, like this. Analyze them under SEM excluded the possibility that the organs have initiated but didn't expand. No. When we see a missing organ, the organ is gone. The primordium is not initiated. So from big, showy flowers like this, we got flowers like this. Same scale bar, 1 centimeter, and sometimes not a flower at all. These phenotypes told us that probably jagged is playing a dual role in Aquilegia flora development, that it promotes distal growth, which is a function that's already known from other taxa, probably conserved. But also it's a key player in primordia initiation, that this role is epistatic to the latter, to the former. Previous studies of jagged in other taxa didn't really reveal any leaf phenotype, but the show is just starting for us. So here we have the Aquilegia wild-type leaf with a trifoliate compound leaf with different segments. First of all, we saw a lot of severe lamina reduction. Sometimes the lamina is so reduced you cannot see it with your naked eyes. Secondly, we saw a lot of weird concave curvatures on our leaf surfaces. When coupled with this severe lamina reduction, the whole leaflet segment forms this kind of weird, kind of cute, bow-like structure. If we take a look at these guys under SEM, they are leaflet as well. The segments has initiated, they are concave, but they cannot expand. If we analyze some epidermal cells of jagged silence leaves compared to wild type, for the adaxial side, we didn't really see any difference in cell shape or cell size. So this told us the severe lamina reduction must be due to a huge reduction in cell numbers. While for the adaxial side, we saw a cell shape change as well. So the wild-type puzzle-like big cells become small and conical, indicating the concave curvatures we see in leaflets could be due to some kind of improper differentiation of adaxial cells. So suddenly, polarity is involved. Well, while we are pondering about this, we saw something even more peculiar, that we saw a lot of outgrowth on our inflorescent stems. To our knowledge, this kind of phenotype has never been reported anywhere in other systems. So I did a histology of these stems. Compared to wild type, the same developmental stage, the same batch of histology. One of the most obvious things we see is that our stem didn't have much xylem. When the xylons do appear, you can see the vascular bundles do have the correct polarity, but there is not that many xylons. When xylons do appear, they are not well differentiated. They are not well lignified. In addition, although most of the bundles show correct polarity in the stem, it's not true for guys near where the outgrowth starts. Usually, on one side of the outgrowth, we'll have a cluster of vascular bundles that look so confused. They are not well developed at all. They are not well positioned. But on the other side, we have more or less normal bundle. Then the outgrowth starts, and there's bundles inside as well, but they are having a leaf-like polarity, not stem-like polarity. What does this mean? Please let me remind you what I talked about in the beginning. Here's what we think. So in wild type, everything's strong, jagged, adaxial, abaxial, jagged. Everything's normal. If we have a defect in the adaxial identity gene, what we expect to see is radialized leaves, and the vascular bundles inside of the stem will change their polarity as well. But this is not what we see. We think jagged affects cells, both adaxial cells and abaxial cells, but it disproportionately influences cells with adaxial identity. So the cells have their identity established, but they cannot respond to it properly. So we got super reduced xylon and concave curvature in the leaflet. However, because of VIX, we don't always get strong and even silencing. So when groups of cells are experiencing weak silencing, cells there will differentiate, proliferate, and differentiate normally to a degree. And there will be where the lamina outgrowth starts, because juxtaposition of adaxial and abaxial identities leads to lamina growth. My name is Mia. I'm from Professor Elena Kramer's lab at Harvard University. Thank you very much.
How do you intend to test your hypothesis?
For the identity-specific proliferation program? Well, so because we know now jagged play a pretty strong role in both flower and leaf development, for our tools available right now couldn't really do anything. So in an ideal world, if I can do whatever I want, I will probably dissect the promoters first and find the part that only affects vegetative tissue polarity, and then mess around with that.
All right, we have time for, we have 2 more minutes, so if any more questions, feel free to ask.
Yes, please.
What are the earliest stages at which you look for this effect?
So the leaf phenotype is completely a surprise. I only saw them when they are shown like it. But for flower phenotype, I think for the earliest one we can see when they are really early, when the flora meristem is still there, when carpels are not initiated yet.